Hello all. In this part, we will study about antibodies and the vaccination and immunization. We will also study about the active immunity and the passive immunity. So, let's start about the first, what is the structure of the antibody is there. Okay, you know that now what is the antibodies and what are the antibodies. Antibodies are the protein molecules that is called also called the immunoglobulins, and the various types of the antibodies are present, like the immunoglobulin A, immunoglobulin M, E and G. Such type of various types of the antibodies are present, and IgG is also there, which is called the immunoglobulin G. Each antibody molecules has 4 peptide chains. 2 are called heavy chains and other 2 are the short, that is called the light chains, and both are arranged in the shape of Y. You can see here, this is the structure of antibody. This structure of antibody present in the Y shape, and hence an antibody is presented as H2L2. That means 2 heavy chains are there and the 2 light chains are there. These antibody are very important in all living organism, because only the antibodies are important to against the immunity or against the foreign particles. Okay, so if we see this structure of the antibodies, so in the structure of the antibody, you know that what is the primary response and what is the secondary response. The primary and the secondary immune response are carried out with the help of 2 special types of the lymphocyte present in our blood. First is the B lymphocyte and second is the T lymphocyte. The B lymphocytes produce an army of the proteins in response to the pathogens into our blood to fight with them, and these proteins are called the antibodies. And what is the work of the T cell? The T cells themselves do not secrete antibodies but help B cells to produce them. Each antibody molecule has the 4 peptide chains. You know that now 2 small chains are there and 2 heavy chains are there. So that's why we are calling like H2L2. The different types of the antibodies are produced in our body, like IgA, IgM, IgE, IgG are some of them. A cartoon of an antibody is given here. You can see here, this is the simple structure of the antibody, and different because these antibodies are found in the blood. The response is also called the humoral immune response and the cell mediated immunity. You know that in last part we have already studied in the acquired immunity. So this is all about the structure of the antibody. The graft from just any source, an animal, another primate or any human beings, cannot be made, since the grafts would be rejected sooner or the latter. And tissue matching, also there is the blood group matching, are essential before the undertaking any graft or the transplant. And even after this, the patient has to take the immunosuppressant all his or her life. The body is able to differentiate self and non-self. The cell-mediated immune response is responsible for the graft rejection. This is about the passive, but this is during the transplantation, and why is it that organs cannot be taken from body, and what is that the doctors check. Okay, so they check here this about the tissue matching or the blood group matching. Okay, so this is about the structure of the antibodies. Next to the structure of the antibodies, on the basis of the production of antibodies, this immunity further categorized into two types. First is the active immunity and second is the passive immunity. Now what is the active and the passive immunity here? The active immunity means the body produces its own antibody against the antigens. That means body have itself ability to produce the antibodies against the antigen, and antibody only will produce when there is an entry of any antigen. So when a host is exposed to an antigens which may be in the form of living or the dead microorganism or the other proteins, antibodies are produced in the host body, and this type of immunity is called the active immunity. That means that is not the artificial immunity, that is the natural immunity present in the every living organism. And the active immunity is slow and takes time to give its full effective response. The injecting the microorganism during the immunization or the infectious organism gaining access into the body during the natural infection that induce the active immunity. This is about the active immunity. And if we talk about the passive immunity, the passive immunity when the ready-made antibodies are directly given to protect the body against the foreign agents, that is called the passive immunity. That means if some infection is happen to the host, the time in his body or her body there is no ability to fight against that disease, that time the ready-made antibodies are transferred from one individual to the another individual. That means let's take the example of corona here. ओके पहले किसी को कोरोना हो गया है, दैट मीन्स उसके बॉडी में वो एंटीजन के अगेंस्ट एंटीबॉडी प्रोड्यूस हो रहे हैं, एंड द अदर इन्फेक्टेड पर्सन who is now suffering from the corona, the time what is going, the antibody from the first host are ready-madely provided to the another host to fight against the corona. That means this passive immunity is the ready-made antibody that will transfer from one individual to the another one. This is the passive immunity. Do you know why mother's milk is considered very essential for the new born? Because whatever the yellowish fluid, that yellowish fluid is a colostrum that contain the antibodies of the immunoglobulin A, and it is the best example of the passive immunity that is provided by mother to her child. So that's why this passive immunity and it is very all the living organism, that yellowish fluid colostrum secreted by mother during the initial days of the lactation has abounded antibodies IgA to protect the infant. The fetus also receives some antibodies from their mother through the placenta during the pregnancy, and these are some examples of the passive immunity. Actually, the different type of the antibodies produced in the blood that include the immunoglobulin A, immunoglobulin M, then immunoglobulin E. There are lots of types of the antibodies are there, and these type of antibodies are called the humoral immune response due to the presence of blood. Okay, we will call the humoral immune response only if there is presence of a blood is there. And human immune system can distinguish between the self and the foreign molecules or about the foreign bodies. So, sometimes due to the genetic or the unknown reasons, the body attacks self cells, and this result into the damage of to the body, that is called the autoimmune diseases. Like in body, sometimes it is autoimmunity is there because of unknown reason or due to the genetic we can say genetic damage is there. The time body have the ability of self attack of the self cells, and because of that self attack of the cells, that result in the damage to the body, and that is called the autoimmune disease. So next is the vaccination and immunization. What is the difference between the vaccination and the immunization? Anyone can tell me? The principle of the immunization or the vaccination is based on the property of the memory of the immune system. The memory word is related to your immune system in the vaccination and the immunization. And whatever we will give to the patient, either we are doing the immunization, we are doing the vaccination, that is based on the how the memory of that patient of the immune system. What do in the vaccination? In the vaccination, a preparation of the antigenic proteins of the pathogens or the inactivated or weakened pathogen. Now that is all is the vaccine, and that are introduced into the body of a patient. The antibodies produced in the body against these antigens would neutralize the pathogenic agents during the actual infection. And then the vaccines also generate the memory. You know that B lymphocytes and the T lymphocytes cells are present. That cells of the B cell and T cells that recognize the pathogen quickly and with a massive production of the antibodies. That is the result after the vaccination to the patient. And if a person is infected with some deadly microbes to which quick immune response is required, as is tetanus, and we need to directly inject the performed antibodies or the antitoxin. Antitoxin means what? It is the preparation containing antibodies to the toxin. Okay. So even in the cases of the snake bites is there, then the injection after the snake bites, the injection which is given to the patients that contain a performed antibodies against the snake venom. Okay, that is the vaccination and the immunization. And this type of immunization, that is called the passive immunization. Why it is called the passive immunization? Because we are giving the ready-made antibodies against the snake when that snake bite is there to anyone. The recombinant DNA technology, or the RDT, has allowed the production of the antigenic polypeptides of the pathogen in the bacteria or the yeast. So the vaccines produced using this approach that allow the large scale production and hence the greater availability for immunization. Like the hepatitis B is there, that vaccine is produced from the yeast, and some bacteria are also useful to produce the antibodies against such so many types of the diseases. Okay, so active immunity is different, passive immunity is different. Also there is a difference between the light chain is there, heavy chain is there, and hence the antibody is represented as the H2L2. So this is the structure of an antibody. Then what is the active immunity and the passive immunity, and what is the colostrum that contain the antibodies IgA. And also there is a difference of the vaccination and the immunization, but whatever the immunization is we are given to the patient, or the vaccination, that is called the passive immunization, because we are providing the ready-made antigens or the antibodies against the specific diseases. That time that is called the passive immunization. Okay,